So hi there, my name's Gina Rutherford and I'm currently studying a PhD at Newcastle University aimed at understanding muscle dysfunction in ME/CFS and developing a drug pre-testing system. So briefly I'm going to outline the key areas that I'm going to discuss during this presentation. So first of all, I'm going to provide an explanation of why I decided to get involved in this research project and also my interest in ME/CFS. I'm also going to provide a key overview of my research directions and aims. I'm also going to provide an explanation of what muscle acidosis is and also present data to illustrate muscle dysfunction following relatively low-level exercise. I'm also going to talk about muscle dysfunction as a consequence of impaired energetic enzyme function. I also present the pH sensing system that I utilise in the lab. And finally, the potential to reverse acidosis through drug treatment, and finally, future research directions. So why did I decide to take this PhD, and what is my interest in ME/CFS? Well, as you know, ME/CFS is a highly complex illness, one that I find absolutely fascinating. However, it's still relatively poorly understood. So there is a real need for research aimed at developing treatments that can be taken forward into clinical trials. I also have a background in clinical exercise physiology, which sits well with the background to this subject, and also I'm interested in fatigue and bioenergetics. So what is the aim of my research project? Well, the aim is to develop a muscle cell pH sensing system which can assess the capacity of drugs to normalise bioenergetic function. So what do I mean by that? By normalising bioenergetic function, I mean treating peripheral fatigue. Also to test and identify key therapeutic targets in a muscle cell-based system to assess the capacity of new novel drugs to treat intracellular acidosis and again treat fatigue. Also to improve the flow of new treatments from bench, so in the lab, to clinic. So what is muscular acidosis? Well, the easiest way to explain muscle acidosis is to look at exercise. So during exercise, ATP, which is a high energy compound which is used to fuel muscular contraction, is broken down to ADP, therefore releasing energy and also producing a proton. However, when mitochondrial respiration can match ATP demand, there is no proton accumulation in the muscle. This is because the protons are taken to another part of the respiratory chain where they're utilised. However, as exercise intensity increases, there's an increased reliance upon glycolytic pathways to fuel the resynthesis of ATP. Therefore, protons accumulate in intracellular space. Essentially, protons are hydrogen ions, so an accumulation results in a lowered pH, therefore acidosis, causing muscle dysfunction, and then the perception of fatigue, because acidity in a cell obviously causes damage to enzymes, so the body perceives this as fatigue. Interestingly, patients with ME/CFS exhibit profound and sustained acidosis following relatively low-level exercise, and this is not normal. And this shows that there's a major underlying bioenergetic abnormality associated with the illness. For example, in a study completed at Newcastle, we found that ME/CFS patients follow a repeat exercise protocol of 3 by 3 minutes of maximal plantar flexion at 35% maximum voluntary contraction, resulted in a significantly lower end exercise pH and a significantly prolonged time to recovery from acidosis when compared to a healthy control group. And this again suggests there to be a major underlying bioenergetic abnormality. Also, anaerobic threshold, which shows the maximum capacity of the aerobic system, was significantly reduced. And this has a short-term implication of acid generation as a result of the overutilization of the lactate dehydrogenase pathway, so again, the anaerobic pathway. So ME/CFS patients are relying on that anaerobic pathway at really low-level exercise, which in healthy individuals, this is not the case. And that's why in the previous slide I used the runner example, because it sort of corresponds with how an athlete would feel at the end of a race. So what causes this muscle bioenergetic abnormality in intracellular acidosis? Well, it's believed to be the result of impaired function of key energetic enzymes. So pyruvate dehydrogenase complex, or PDC, is an enzyme, a three-component enzyme, which converts pyruvate generated during anaerobic glycolysis to acetyl-CoA, which can then enter the Krebs or TCA cycle to be aerobically metabolized, leading to normal muscle function. However, a second enzyme called PDK, or pyruvate dehydrogenase kinase, a bit of a mouthful, but there it is, but this enzyme functions alternatively to inhibit PDC function. And this can lead to pyruvate being alternatively metabolized anaerobically. So the enzyme lactate dehydrogenase converts pyruvate to lactate, resulting in intracellular acidosis with the end point of, again, muscle fatigue. Interestingly, however, a drug called dichloroacetate, or DCA, has been found to completely normalise muscle function, and it does this by inhibiting PDK, therefore restoring PDC function and overcoming muscular acidosis. The reason I mentioned DCA is because it will appear in a later slide, so I just thought I'd give a brief overview of its function. So how do I go about sensing pH in muscle cells? Well, I utilise a muscle cell-based nanosensor platform, and this enables the fluorescent-based assessment of pH in real time without impacting on the cell function, so that cell function is maintained and so is the integrity. As you can see in the image at the side of the screen, the analyte of interest, in this case pH or hydrogen ions, can enter the intracellular space, well, the nanosensor space, where it interacts with a dye of interest. Upon excitation, normally by a plate reader, the dyes create an emission, and this emission is proportional to the level of interaction between the dyes and the analyte of interest. And this can be, the data is then provided by the plate reader, and we can use that data to measure pH. So how does the nanosensor actually get into the cell? Well, the nanosensor is delivered into the cell by a liquefaction mediated delivery technique. So liquefacted meaning the nanosensors are incubated. They're then added to a plate containing cells. The nanosensor and liquefaction form vesicles which bind to the cell membrane, and the cargo is then released into the cell. So how do I actually use a nanosensor to determine pH effectively? Well, before I can determine pH, I must first create a cell-free calibration. So this involves just dissolving the nanosensors in a buffer solution over a range of pH values, so generally 4 to 8, ensuring normal physiological pH is incorporated. A plate reader can then be used to analyse the samples, and then a graph can be constructed. So on the y-axis we have fluorescent intensity of the dyes, and this corresponds to, on the x-axis, a pH value. And once we've constructed this, we can then use this graph to determine the pH of any nanosensor doped cell. So is there evidence to suggest that the nanosensors do make it into the intracellular environment and that they are pH responsive? Well, preliminary data suggests yes to both. For example, in the images depicted on the screen, nanosensor doped muscle cells were treated with 1 micromolar of lactic acid, and following 30 seconds acidification, you can see that there's fluorescent activity within the nanosensor. However, after 600 seconds, there's been a pH-dependent quenching of fluorescent activity. So this data shows that yes, the nanosensors are getting into the cell, and also that they are responding to acidity. Interestingly, preliminary data also shows that intracellular acidosis as a consequence of impaired energetic enzyme function is completely reversible. So in this graph, we've got the MECFS cells and the non-disease cells. They were obviously treated with nanosensors, and they were treated with dichloroacetate, the drug I spoke about earlier. And as you can see, at baseline, the MECFS cells exhibited significant acidosis compared to the non-disease cells. However, following drug treatment, this was completely normalised. This essentially forms the basis of my PhD because it shows that intracellular acidosis is completely reversible. and therefore fatigue associated with ME/CFS is treatable. However, there's a number, at the moment, DTA can't be administered to patients, as it's associated with a number of quite severe side effects. Therefore, the aim of my PhD will be to use the muscle cell sensing system to assess the capacity of a range of drugs to normalise intramuscular pH in a safe and effective way. So the end goal will be to have a drug, hopefully get it into clinical trials, and eventually be able to give patients a drug that we can take to completely combat muscle dysfunction as a symptom. Another drug target will be lactate dehydrogenase, because obviously there is an overutilisation of the anaerobic pathway, and this is a key enzyme. And we've also recognised a few drugs that can act to inhibit this enzyme. So thanks for listening to my talk. I'd like to thank firstly Action for ME for funding my research project, and also the National Institute for Health Research. Thanks very much.